So I wanted to do a brief reminder that there are a lot of groups in our community that are actively working to help individuals who live with brain injury. And the Whitebird Clinic is one of them. Whitebird Clinic is a collective environment organized to enable people to gain control of their social, emotional, and physical well-being through direct service, education, and community. And part of our goal today is to raise funds for critical resources for the brain injury community, such as Whitebird Clinic. And we highly encourage and request that you consider donating them today. So I'm going to go ahead and put the link to that donation page in the chat. And then Dr. Trooster, I had some individuals asking if the slides that you showed will be available or...
Happy to send it over. And so you just tell me, you can just email me with who to send it to, and you guys could share it to anyone you want.
Okay, wonderful, amazing. Thank you so much, and everyone for being here while we're working things together. And then just really quickly, so Willie Curry is a PhD candidate at the University of Vermont, researching the neural basis for epilepsy at a cellular level using rodent models. And then Willie will introduce you to the famous case of HM and how it helped elucidate the mechanisms in various types of memory. I just wanted to give that brief introduction. And then Willie, the floor is yours.
Thank you so much. I want to thank Daniel first and foremost for inviting me. And I also want to thank Dr. Traster for giving a pretty great talk previously. I'm not sure my talk will have as much utility. It's more kind of a historical overview with a little introduction on what I'm currently doing in the lab, but hopefully you'll enjoy it anyway. I'll get started. So I'll just share my screen real quick. Let's see, there we go. Okay, everyone can see that? Okay, are we good?
I can see it. Can everyone else?
Cool, great, awesome, thank you. Let's go ahead and get through. So yes, hello. Thank you for the introduction. I'm Willie Curry. I'm a grad student here at the University of Vermont, and I study epilepsy. Although this talk is more about so it's entitled HM, The Life of Memory, and it's called that for two reasons. One, that to know the life of this gentleman here, HM, as he was known throughout most of his life, is to know the history of how we came to understand certain types of memory, but also that memory kind of aids us in kind of constructing a sense of self and therefore a life. So with that said, so I want to be fairly cliche and kind of open up with this poem by Emily Dickinson, who's one of my favorite poets. So I'll just kind of cringily read this out. I felt a cleavage in my mind as if my brain had split. I tried to match it scene by scene, but could not make them fit. The thought behind I strove to join until the thought before, with sequence raveled out of reach like balls upon a floor. So, great imagery from Dickinson as always, and kind of gives some kind of tangible kind of depictions of what it might be to have problems with one's memory, feeling very fragmented. So this is HM, or as he was known, or now we know, since he recently passed in 2008, I believe, now we can kind of know his real name. His name is Henry Meliason. He was born in 1946. He experienced a bike accident. He was knocked over by a bicycle at age 7. And after that, he started experiencing seizures. At first, these seizures were what we call absent seizures, in that they were, he mostly retained his consciousness. It usually would kind of, if you were to look at him while he's having the seizure, it's kind of looks like he's kind of blankly staring into the void, just kind of spacing out. But increasingly, by age 16, and then especially by age 27, these seizures had gotten considerably worse, life impairing. He was the kind of things that you might associate with, even though we don't use this terminology anymore, what you might associate with a grand mal seizure, with tongue biting and incontinence and losing consciousness. And he was having these types of seizures several times a day. And I think he constructed engines, but eventually the seizures became too much and he couldn't actually perform on the assembly line. So there is some evidence to suggest that there's a possible genetic component to his condition. Apparently, a few of his first cousins on his father's side had seizure disorders as well, but I don't think that's been fully explored. So by the time he's 27, he reaches out, or really he was approached by a surgeon who I'll mention in the next slide, a guy named William Scoville, who carried out a particular type of surgery to excise certain parts that he thought was causing the seizures. And this surgery was carried out in 1953. So just a brief aside, this is oops, my bad. So a brief aside, just to talk about temporal lobe epilepsy. This is a slide I use in my talks introducing my work. Epilepsy affects 50 to 65 million people worldwide. It's depending on the list you refer to, it's the second or the third most common neurological disorder behind stroke and TBI. It is the most common form of epilepsy, presenting in 30% of people who have epilepsy. Its complications include stigma because it's kind of characteristic symptom seizures are really kind of bizarre and strange to people who don't experience them. Along with the seizures, there's psychological dysfunction, specifically in the realm of working or short-term memory, and this could lead to a decreased quality of life. So currently, the treatments in the clinic are to treat the seizures. They're the most kind of devastating and visible part people know about or component of the epilepsy, but that can de-emphasize the cognitive deficits that are there as because of the etiology of the disease, but also a lot of the anticonvulsant or anti-epileptic drugs that are meant to control seizures also come with their series of adverse side effects. So that's not good. So what I'll try to show later is my work involves looking at alternative treatments for types of epilepsies, specifically temporal lobe epilepsy or TLE. So these pictures on the right here are just to show you that when we talk about the brain, despite what Dr. Trasser said earlier about the brain being an incredibly complex and kind of multi-parallel system where sometimes things aren't as modularized or kind of in specific dedicated spaces as much as we'd like. But despite that, we kind of put things in these kind of structural functional regions called lobes. There's a frontal lobe, as the name would suggest, that's at the front. And then there's parietal, occipital, and temporal lobes. And the temporal lobe is this kind of green portion here that contains these structures that are believed to play a huge part in temporal lobe epilepsy. And those components played a part in the symptoms that HM was experiencing. And then this picture at the bottom here, these are kind of, we just kind of cut the brain in half and showing those structures that are there in the temporal lobe, the hippocampus, which is an incredibly important and well-studied, what's the word I'm thinking of? Structure, Jesus. A well-studied and important structure for the formation of memories, along with a number of different things. The amygdala is there, a structure that's hugely important in emotion and motion regulation. Then there's other things like the parahippocampal gyrus that also plays that kind of supplements the functions of those other structures. So to go back to HM, so 1953, he meets up with William Scoville at, I believe, Hartford Hospital in Connecticut. And this is kind of a diagram of what was actually done to him. So Scoville went in from the front, basically took a kind of suctioning device, a small vacuum, and vacuumed out certain portions of the temporal lobe. Here it says roughly about 5 to 8 centimeters. I think it's closer to 5. So actually, I'm going to keep this in mind. I'm actually going to go to this slide, which might give you is a little more visible. So here on the right side, or excuse me, on the left side here, we have, if we're looking at the brain from the front and we just take a slice, there's a hippocampus that I mentioned earlier, this structure that's really important for memory. And it's hanging out there, and that, at least part of HM's hippocampus was removed, along with the hippocampal gyrus, which is a supplementary area. And if we move over to the picture on the right, this actually is HM's brain after it was removed and fixed, but before it was sliced, and there was a pretty deep analysis of what his brain looked like. But you might be able to appreciate in these boxes, A and B are, this is where those structures were they previously were there, but then they've been sucked out. If we were to look at a normal brain, these areas here would be covered with more tissue. So, okay, so HM has the surgery and it's a success. He stops having these debilitating, life-impacting seizures, but at a bit of a cost. So we learned through Brenda Milner is a researcher who comes and along with Scoville studies HM for a number of years. And what we find is that HM has, so up until a certain point, he can't, so actually, excuse me, let me back up. So let me actually refer to the diagram that I have here on this slide. So here is the moment that HM experiences his surgery. So it seems like he is missing some memories, mostly 18 months, I believe, up until the surgery, but then complete loss of memories up 18 months prior to his surgery, and then sporadic memory loss of memories 3 years prior to the surgery. Before that, he remembers perfectly everything. He can remember his childhood, he remembers going to school, whatever. So we call that retrograde amnesia. Retro, as the name would suggest, is forgetting things that happened to you already before in the past. But also HM very demonstrably experienced interrograde amnesia. But it wasn't full scale. So it seemed like HM could form short-term memories, and I'll elucidate what that looks like specifically in the next slides. But he couldn't form long-term memories. Okay, so what does that mean? So if you're talking to HM post-surgery, he could carry a conversation, but he immediately forgot the people associated with, or the person he was having a conversation with, and the contents of that said conversation, especially if he was distracted after it. Kind of going to specific things. There were, he could remember 3-digit numbers up to 15 minutes, but with sustained attention. And again, if he was distracted, he would remember faces, or he would forget faces and events in less than a minute. And also, which I think was more of a problem for him later in life, as opposed to earlier, is that he had an impaired ability to respond and rate pain, and also hunger and thirst. He reported no difference in satiety or how full he was, both before and after eating. So one particular task that Milner had HM do was this kind of 5-pointed star task. So he had to trace the outline of this 5-pointed star in a mirror, but he could only see one of his hands. So it was kind of a test of motor function and to see if he could increasingly get better at doing it over time. His motor skills were a little delayed. It turns out that one of the anticonvulsant drugs to treat seizures, phenytoin, which he'd been taking for a number of years, actually resulted in a bit of atrophy of his cerebellum, which is really important for balance and coordinated physical movements. But anyway, so he takes this 5-point star task. He does 10 trials over 3 days, and he gets better at it. But he doesn't remember doing it those previous days. So that kind of opens up, as I'll show you in my next slide, that kind of opens up this idea that there's not a kind of overarching memory, so to speak. There are independent, but also interdependent memory systems. So kind of moving from the kind of, memory is a complex thing. We go from, Socrates talking about anamnesis, which is like kind of, no one really like learns anything. It's just remembering things that you already had because the soul's immortal or something. So we move past that. We get into a breakdown of immediate, what seems very like intuitive and obvious to us now. There's both immediate and long-term memory, which has been discussed since the late 1800s with psychologists like William James picture. But then then studies of HM show that there's also this thing that kind of bridges both immediate and long-term memory called declarative, and there's this thing called non-declarative memory or procedural memory. So HM was lacking one, but not the other. So he's lacking declarative memory because the regions that are associated with that, such as the hippocampus, the perihippocampal gyrus, amygdala, those were the parts that were partially removed during a surgery, but he still had his non-declarative procedural memory intact, which are things things like basal ganglia, the cerebellum, although in HM it was a bit atrophied, and the neocortex. So it seems like, so now we think that there are certain, so you can learn how to, the mechanisms by which you learn a skill and get better at a skill are not necessarily tied to things like autobiographical memory or your ability to remember actually having done the skill, which is incredibly interesting. Actually, never mind, I forgot what I was gonna say. But even then, it turns out that the story is not even that simple. There's a bit more nuance. So Suzanne Corkin is another researcher who also studied HM throughout most of his life up until his death in 2008. And there were a few quirks to his ability set. So everything I said, previously holds true, but also there were things like, so HM had his surgery in 1953, and him and his family moved into a new place 1958, and they stayed there until 1974. So Dr. Corkin had HM draw the floor plan to the house that he moved in 1958 in 1966. So this would be, and it also turns out that he was able to roughly draw the same floor plan in 1977, I think 3 years after he had moved out of the home. So despite not having the ability to hold kind of long-term autobiographical and declarative memories, for some reason, he was able to remember, he could draw the floor plan of a house that he moved in post-surgery, and they left, and even after he left it sometime. And then on the right here, it also turns out that when primed for certain famous names, even names of people who weren't famous until after HM had a surgery, with a bit of priming, he was able to kind of produce the names of those people. So that's very interesting. And the reasons behind that are still not sufficiently clear. So there's more kind of thinking and research needs to be done about that. So that kind of wraps up kind of HM story. And I just very briefly want to close out with look at my research, which does address memory. So this is what I do. So basically, I take stem cells. I take a type of stem cell, and I implant them specifically into the hippocampus, the area that was partially, or one of the regions that was partially removed with HM. And I take stem cells, I implant them into the brains of rats that are epileptic. There are a number of different, both genetic and acquired rodent models of epilepsy. So I put those in the hippocampus. I give those cells time to kind of mature and grow and kind of integrate into the hippocampus. And then I do behavioral experiments. I have these animals. I'll explain what I do in just a second, but I have them do behavior. And then I think, as Dr. Traster referred, I use microelectrodes. So I have these little bundles of wire. I put them in the hippocampus, and I record the neural output. I can record the electrical activity of the neurons directly, and then I can analyze what those data mean. So this is just a picture of what. So I take these stem cells, I color them green, and then I stick them into the hippocampus. And this is just a kind of representative picture of what my graphs look like. So
where do you usually implant? What parts of the hippocampus?
So I have 2 injection sites. So I basically do the hilar region, like in between the wings of the dentate gyrus, and then I try to aim for CA1 as well. So and both of those are going to be in dorsal hippocampus, where you see this particular type of layout as opposed to the ventral portion. So with that said, so yeah, I do behavior, and let's see, so I do something called the Morris water maze. It's this very standard task of kind of working memory, but also what's known as spatial cognition. For years now, neuroscientists and psychologists have theories about how animals, including humans, obviously, know where they are in space in an environment. There seems to be certain types of cells in the hippocampus that change their firing rate depending on where an animal is in an environment. We call those place cells, and I won't harbor on those too much. But anyway, in order to test an animal's spatial cognition, something we do is the Morris water maze, where you fill a big tub of water your big tub full of water, and then you gently place the animal at certain points along the perimeter of the tub, and you have a hidden platform, that's right below the surface of the water, that they can get onto, but they don't know it's initially there because it's obscured. Rats are really good swimmers, but they don't like to stay wet for any particular long amount of time. So this platform is an escape for them to get dry. And as you put the animal, animals that are just kind of normal, don't belong to any kind of experimental group or anything, they get better at the task over the course of a number of days. So I have 3 groups. I have control animals where no treatment's been given. I have the seizure animals. They're polycarpine animals because that's the agent that I use to induce seizures. Then I have MG cells. That's the cells that the stem cells that I'm using, and I put them into the epileptic animals. And it seems like, so just to kind of give you show you what this graph means. On the y-axis, just the proportion of animals that reach the platform. They get there, and they're given 120 seconds to do so. So all this graph is just showing you is that this is a survival curve. You might be familiar with it when plotting outcomes for, say, cancer treatments, but we're kind of doing it in reverse. But the control animals, perform just fine. But at least as far as the data shakes out so far, the animals that get the stem cell treatments perform a little bit better than animals that don't. And then I'll just kind of, so within the hippocampus, so I haven't really touched on it, and I think Dr. Berry will give you a bit of an overview, I think in the next talk, or maybe 2 talks for now, is that the hippocampus functions the way it does because there are so-called brain oscillations that are there. And one in particular that's really interesting and necessary for function is the so-called theta oscillation. And cells in the hippocampus like to fire at specific points in accordance with the theta oscillation. And this graph is just showing you there's differences, the nature of how well they fire in accordance with theta. And it seems like there are demonstrable differences. There's significant differences between the epileptic animals and versus the control and animals that receive the stem cell treatment. So with that said, conclusions are just that memory consists of a complex series of interconnected systems. It's its own distinct phenomenon, removed from other cognitive and intellectual functions. Deficits in one domain don't necessarily carry over to others. There might be systems or network level mechanisms via the kind of oscillatory activity to explain whatever improvements and deficits, for instance, that I'm seeing in my animals. And there are, of course, things that we still don't know and we're continuing to work on. So yeah, that's it. That's and that's all I got.
Thank
Thank
you
you, Willie.
so much.
I think that's a good point you made in your conclusion there. Memory is distinct, although there is some overlap and connection with other brain regions. It's a very specific function.
Yeah.
Cool.
Wonderful. Thank you so much for sharing.
Yeah, no, if anybody has any questions, I know I kind of, and that's all I got.
Thank
Thank
you
you, Willie.
so much.
I think that's a good point you made in your conclusion there. Memory is distinct, although there is some overlap and connection with other brain regions. It's a very specific function.
Yeah.
Cool.
Wonderful. Thank you so much.
Because it's usually when you dig into the mechanisms, how epilepsy even comes to develop, what's known as epileptogenesis, there's just a variety of factors that aren't even necessarily well, they're studied, but there's not a particular mechanism that is standard and people say, oh, that's the thing that caused it. So I think there's probably a correlation between the number of brain regions involved in that temporal lobe area, because there's just there's amygdala, there's perihippocampal gyrus, there's hippocampus. I think it's probably a function of the proportion of regions that are affected. That probably has to do with, so if you're damaged, if a damage from a particular traumatic brain event is sequestered to just the hippocampus, that might be not reaching a particular threshold that's needed, as if other regions were incorporated as well. Does that make sense?
Well, I'll just basically call what you just described as there's an element of a moving target in all of this. for lack of a better way of putting it.
Yeah. So, and you never really know. So the unfortunate, I mean, it's the brain, so it's really
Yeah.
kind of hard to get at. So,
Right.
I mean, and it's a complex system. So
Right.
oftentimes you won't know, and I guess much like Alzheimer's, like you won't really know the extent of the damage that led to a particular outcome until you do postmortem looks at the brain region. So,
Okay. Thank you.
yeah, no problem.
Wonderful. Thank you so much for sharing.
Yeah, no problem.
And then just as much, if you are willing to share your slides with us, you can just email
Mhm.
us the blank and then we can distribute it out to all of the attendees.
Sure. Yeah, we'll do. Will do.
Wonderful.
Yeah, thank you, Willie. Thank
Yeah, no
you.
problem.